All right, so thanks for the introduction, and I really appreciate being able to participate in MetroFlow. It's been a few years since I've had the opportunity to attend. As you know, ThermoFisher is a world leader in serving science, and we offer solutions for the entire workflow for flow cytometry. And today we will be focusing on some of our reagent solutions. And this is the agenda, and it does seem fairly full. I'm focusing on just some of our newer solutions. And so I have about 2 minutes per topic. So I'm just gonna kind of go through this very quickly. So we have new antibody conjugates in the brilliant ultraviolet 737, 805, and Alexaflor 561 and Alexaflor 660. Yes, ThermoFisher does now sell the BUV 737 and 805 dyes, as well as conjugates of the 660 and 561. These are fairly bright dyes to help with lower expressed targets, and can be useful in both spectral and conventional flow cytometry. Now, I'm not going to go into the details of what BUV dyes are. I think we're all fairly familiar with them. We do now have around 80 new clones of BUV 737 and around 60 new clones of conjugates with a BUV 805. Now, we also offer the Brilliant Stain Buffer, as well as the Super Bright Complete Staining Buffer. And of course, you need to use one of these if you're going to use more than one polymer dye to mitigate the polymer to polymer interactions. And since this is a session on spectral, here are some spectral plots. These are from the 5-laser Cytec Aurora and the 7-laser Bigfoot spectral cell sorter. Now, of course, they're from different instruments with different configurations, so they won't be identical, but it's just nice to take a look at what some of these spectral signatures do look like. So the Alexaflor dye series is well established and known, and now some of the dyes like Alexaflor 561 and 660 are looking at greater value in flow cytometry as we enter the spectral world. So here we see some of the details around the dyes. These are fairly bright, so they can be used with those dim expressing dyes. And here at the bottom, we see some of the Aurora spectral signatures. When we start looking at spectral, it's interesting to look at how similar and how different some of the dyes are. And here you can see in this particular similarity comparison that Alexaflor 561, it could potentially be used with PE. I would suggest that if you do that, you put those on mutually exclusive targets. And Alexa 660, it can potentially be used with APC and Alexaflor 647 if you're looking to make a fairly large panel. We have some other options. You have probably already heard of the NovaFlor dyes. I know MetroFlow has had a number of presentations around these in recent years. These are unique spectral signatures and designed with narrow emission profiles. So basically, this is a new type of foundational dye. It's using this phyton label where this is small molecule dyes are precisely positioned on single-stranded DNA. And these assemble into this remarkably stable DNA structure, which is called the phyton. And the fluorophores are placed at optimal distances to engineer frat efficiencies. So this allows for a different choice of these small molecule fluorophores and allows for a variety of different excitation and emission combinations. So the NovaFluor dye portfolio currently has 19 different colors. You can see them all listed here. So the naming convention is the laser that is the primary exciting laser, like blue, yellow, and red. And then the number is the peak emission. Now we do have antibody conjugates with all of these. And just this month, we have released some new clones with a number of these dyes. So we are aggressively expanding content with these dyes. There are conjugation kits and custom conjugation services as well. So each vial of a NovaFluor antibody conjugate comes with the cell blocking buffer. We do recommend the use of this buffer with all of the NovaFluor conjugates when labeling cells for background reduction. You do not need to use it if you are labeling antibody capture beads. The blocking buffer can also be used with any conjugate as a monocyte and macrophage blocking solution. So just in summary, with the NovaFluor dyes, this is a new foundational dye with unique spectral signatures, designed with narrow emission and minimal cross-laser excitation. And this should help with higher resolution in your multicolor panels. So moving on to Amine reactive dyes. Again, this is a dye type that I'm sure you're all familiar with and use on a regular basis. We do have 6 new colors in this dye series, and you can see them highlighted here in the total number of 14 that we currently offer. And again, the naming is, the emission is in parentheses, and then at the end is the primary laser line. So looking at the 6 new live dead fixable stains, again with the Bigfoot and the Aurora spectral signatures, you can see a little bit of how they perform in those particular instruments. And I did want to mention the Fluorescence Spectra Viewer. This has been recently updated with improved ease of use and greater functionality. So if you have been in to use the Spectra Viewer recently, you've probably noticed some of these changes. And for those of you who haven't, the next time you go in, you can look at some of the changes. And if you want to look at more, there is a Spectra Viewer guide that you can use to look at that. And then finally, we do have some spectral systems now in the flow cytometry panel builder. So in the drop-down menu, there are Aurora and Bigfoot instruments listed, and the panel builder identifies these as spectral instruments. You'll notice when you get to the fluorophore selection page that you will see that the spectra are displayed at the top. There is a complexity score and a comparison matrix. When you get to the step of pairing antibodies with fluorophores, the choices will be listed in order of complexity impact. And as the fluorophores are added, the complexity is tallied up. And then this is a view of when you click on the comparison matrix. And this shows the relative similarities of the selected fluorophores in the panel. Values close to 0 mean the signatures are very different, and values close to 100 indicate that the signatures are very similar to each other. And there is a hover feature where you can hover over one particular section and see what the dye combination is for that particular combination. And with that, I will thank you for your time. And if there are a lot of questions, I really encourage you to talk to your local ThermoFisher rep, and they can help you with more of any of these new dyes.
Thank you, Jolene. That was fantastic. There's a lot of cool stuff you guys are doing.
Yeah.
So also there are some questions, but again, for the interest of time, I think we'll save those to the panel discussion that will happen after this next talk. So
Okay.
Thank you again.